So we're looking at biotechnology, and remember, one of the big things that you need to be able to do is directly apply your knowledge of DNA and the unity of DNA across organisms to the problems of biotechnology. So you need to be able to understand what a restriction endonuclease is and how it works. You need to understand what a gel electrophoresis is and how it works. You don't necessarily need to understand the idea of alkali solution or exactly what nitrocellulose is. But you do need to understand that if we're trying to separate and identify segments of DNA, that you should be able to talk through the tools that you would need in order to do that and what exactly those tools do. So this first part of biotechnology was really looking at how we just identify segments of DNA. We cut them with the restriction enzyme into different pieces. Now keep in mind, if we're looking at different organisms, let's say we're looking at the Escherichia bacteria, and then let's say we're looking at Neisseria gonorrhea bacteria, they're going to have different DNA sequences, and they're going to be cut in different ways. So while there is unity around DNA, while all living organisms have DNA, and it has a sugar phosphate backbone, and et cetera, while there's unity, there's also diversity in the exact sequence. So those are things that you know, that you've learned, that you need to be able to apply. So this first part, we're trying to isolate and identify. We're trying to pull out certain sequences. And of course, when we get down here to talking about the probe, probes are going to be used a lot in biotechnology because they want to see if they actually got the right gene. They want to see if the gene is actually active. So this is another reason why you need to understand complementary base pairs. This is why you need to understand radioactive isotopes. So this idea of how we can actually see if something is working or not. Although your notes ask you to summarize the steps of each one, that is an important tool to help you study and understand this. But the bigger key is seeing the big picture. What are the specific tools that we need to be able to use? The AP exam is not going to ask you to describe the steps of southern blotting, but they might give you a scenario where a single piece of DNA is absorbed through a gel, and they might want to know why some pieces of DNA are absorbed faster than other pieces of DNA. Knowing that restriction enzyme cuts them into different size pieces, that's how you begin to really apply your knowledge. We're going to move on to talk about the idea of recombining foreign DNA. And so if we can cut out pieces of DNA, we can go back to one of the things that we said at the beginning. Well, what do we really want to do with biotech? We might want to replace a defective gene. We might want to create an organism that has new genes to itself. So this second big idea is that we can recombine foreign DNA. And so there are a few vocabulary words that we need to understand. The first is the word recombinant, which sounds and looks a lot like the word recombine, and therefore means a very similar thing. Recombinant DNA is multiple sources of DNA. Foreign DNA plus the host genome or the host DNA. So if I want to use gene therapy and replace a defective gene, I'm going to have the host DNA, and then I'm going to put in foreign DNA that wasn't there before. Once I have host and foreign DNA, that's called recombinant DNA. Once we have an organism with recombinant DNA, we actually have what's called a transgenic organism. So an organism with recombinant DNA is called a transgenic organism. Trans meaning across, DNA from somewhere else. And so these are two pretty important words. But now, this is a grand idea. How do we actually implement it? How do we make an organism that has genes that it wasn't born with? It didn't directly inherit. So we know that the main steps are going to be that we need to locate and isolate the desired gene. So we need to go back to our DNA. And the way that we locate and isolate desired genes has a lot to do with probes, being able to find the gene, and a lot to do with endonucleases or restriction enzymes. Endonucleases or restriction enzymes. So we go back to the restriction enzyme, which we already know is going to cut or cleave the DNA. However, the key is that it's cutting or cleaving DNA at a specific site. So HIND3 is from Haemophilus influenza. That's a bacteria. This is the third restriction enzyme from them, but it has a restriction site. So this area right here, AA, GC, TT, this is the restriction site. And this is the only location that this restriction enzyme will cut at. Not only will it cut there, but it will actually cut there in a very specific way. And so you notice this idea of sticky ends versus down here, blunt ends. The advantage of sticky ends is that sticky ends can complementary base pair. With another sequence. So this is how we build recombinant DNA. Right? If I want to cut and paste something, we do it on the computer all the time, we do it with scissors. What are the tools that we have to do this in our cells? Well, the endonucleases will cut, and then we actually have an enzyme that will seal things together. DNA ligase. So we're actually going to cut out desired genes and put them into the host cell. So the first thing that we need to do is locate and isolate the desired genes. Okay, check. We've done that. We found the gene that we want, and we've actually isolated that gene. If you go back to the steps about, you know, how to use the nitrocellulose paper and the probes to actually find a specific sequence that we want. So we have the gene, but it's not in the organism yet. And so what we actually have to do is we have to use the same restriction enzyme on the desired gene and the host genome. Remember, our second step that we're trying to do is we're trying to incorporate the desired gene, into the host. Well, I can't just put the gene into the cell alone. Otherwise, that will just be cut up by the cell and degraded by the cell. I need to actually get the gene into part of the genome. And I need to get it near a promoter. So there's a lot of steps here that have to go on that we're going to kind of skip over. How do I find a promoter? How do I make sure that I'm not cutting the gene in the middle of the gene? We don't want to cut in the middle of the gene. We want to cut far ahead of the gene and far behind the gene. And if we cut into our host, we don't want to cut into our host in the middle of a needed gene. So of course there are all these difficult things that a biotechnologist would actually have to consider. For now, we're going to look at the basic steps. 1, how do you find the gene? And 2, how do you incorporate it into the host? And so we have the basic idea that if we cut them with the same restriction enzyme, then I have these sticky ends. And voila, they base pair together. I need my ligase, and they'll be joined. Except we have a little bit of a problem in that how do we actually get it into the cell, and how do we actually get it into the nucleus? So one of the big things that we're going to have to look at is a delivery vehicle. And these delivery vehicles are called vectors. So we have this big idea of a vector is needed to deliver the gene. Now there's one type of organism that we mostly use in biotechnology, and that's bacteria. And so the vector that we use for bacteria is called a plasmid. You already know that a plasmid is an extra chromosomal piece of DNA. The bacteria are used to having plasmids, and so we can splice open the plasmid, put in the gene, and voila, it's now in the bacteria. However, if all we've done is get one copy of the gene, we don't want to do this to one bacterium, we want to do it to many. So while we need something to deliver the gene, we actually also need what are known as extra copies or clones of the gene. And we're going to use something called polymerase chain reaction, or PCR, to make extra copies. And that should make sense. We know that polymerase, DNA polymerase, will make copies. Now we're actually able to do this in the lab. We're actually able to go through a couple of steps to make lots of copies of a gene. And so I need to give you those steps. The first is that we use heat to denature. Now remember, when we denature, we're not actually breaking the main bonds, we're just separating the two sides. So we denature and we have our left and right strand. Then we need polymerase, so we need to add the enzyme polymerase to actually perform this function. However, this is a pretty warm temperature that we're at to keep them denatured, so we're actually going to need to use polymerase from a heat-tolerant organism. Interestingly enough, they got it from a bacteria that lives in a hot spring. And so we're actually having to solve all of these problems in biotechnology. And then the third thing that we need to be able to do is we actually need to add primer. Remember, DNA replication only happens on a primer that already exists. So polymerase chain reaction is heat it, separate it, add primer, copy. Do it again, do a chain reaction. Heat it, separate those new copies, add primer, make more copies. And so we make many, many, many copies of our desired gene. We put it into a vector, and then we can put it into a bacteria. Interestingly enough, when this recombinant bacterium reproduces, what do you think the offspring will be like? If everything in this cell gets copied, the brand new gene will get copied too. And as long as the gene is put near a promoter, then the enzyme will actually be able to perform protein synthesis. The last thing that we just want to be able to remind ourselves is, what are the actual applications of this? This picture gives you a lot of different applications. Genetically modified plants for pesticide resistance, genetically modified bacteria for cleaning up toxic waste, making human proteins like insulin or human growth factors. We do want to remember that eventually we would one day like to be able to perform gene therapy, but this becomes really complex when you talk about a human that has a nucleus, a human that has many, many cells. So we start to see a lot of concerns about, are we doing the right thing by performing all of this technology? Things to think about.